last time we finished transcription, so now we're going to move on to translation. So when you look at this slide, you need to know, why do we call the mRNA mature mRNA? So what's the difference between pre-mRNA and mature mRNA? So pre-mRNA is the end product of transcription, but the pre-mRNA has to go through some modifications, right, in order to be the mature mRNA that can exit the nucleus and deliver the information to ribosomes. So there are 3 major modifications. You remember the 5' guanosine triphosphate cap, right? So we're going to say 5' cap, and then there's the 3' poly A tail, and then we also need to remove the introns, right? Splice out the introns, because the introns are non-coding sequence, so they're not going to be useful in terms of translation, so we need to remove them. But due to introns, we could have alternative splicing, right? So the same gene sequence can actually generate multiple possible protein products, or really polypeptide chain products. Okay, so once these modifications have been completed, now we have the mature mRNA. So mature mRNA is gonna leave the nucleus and they're gonna enter cytoplasm. Okay, so this is where translation takes place. Remember, transcription takes place in the nucleus, but translation occurs in cytoplasm, and that's because the ribosomes are in cytoplasm. Alright, so the genetic code has been discussed in the online video that I found for you guys. So just real quick, you need to remember that the codon has to have 3 bases, because if there's only 1, that's only 4 possibilities, right? That's not enough, because we need to code for 20 amino acids. So we need a number that's greater than 20. If we have 2 bases, that's not going to work either, because there are only 16 unique combinations. But if we have 3 bases, that's a 64 codes, right? 64 different combinations, which is plenty for the 20 amino acids that we need to code for. So in this case, you may have multiple codes that will code for the same amino acid, which is fine. So this is the codon table. There's a 1 codon you absolutely have to memorize, okay? And that's going to be AUG, the start codon. So you have to memorize AUG is the start codon, and that also codes for amino acid called methionine. And you also have 3 stop codons. So over here, UAA, UAG, and UGA. And then the rest of the codons will code for the real amino acids. And you can see that a lot of times you have multiple or more than 1 codon that codes for a particular amino acid, right? So for this one, the 4 codons are the same for glycine. And then we also have, these 2 for glutamic acid. So you have a little bit, almost like a wiggle room. All right. So we are going to move on to this question. Now, this is a really hard question. I forgot that I had some twists into this question. So let's just kind of go through it step by step. So this is a DNA template. Okay? So we're going all the way back to transcription. So if a sequence for DNA template is given, you need to first generate the mRNA sequence, right? And then you are going to use the codon table to figure out what amino acids are coded from the information in mRNA. Okay, so let me erase that. Okay, so this DNA template goes from 5' to 3'. But we cannot read the sequence from 5 to 3, because remember that, let me use a different color. Remember, the RNA polymerase reads the DNA sequence from 3 to 5, right? So that the new RNA sequence or the RNA strand can be grown from 5 to 3. So if I give you a DNA sequence from 5 to 3, you need to read it not from 5 to 3, but the other direction, from 3 to 5. So this ensures that the mRNA sequence goes from 5 to 3. Okay, all right, so let's read it. So this is the sequence for mRNA. We're gonna read it from this end and backwards. So we have A in DNA. So what's the base that pairs with A in RNA? Is it T? No, it's U, right? Remember in RNA, T is replaced by U. So A pairs with U, and then the next is also A, so it's also U. So U, U, G. All right. Next one, C, A, U. All right. Next one, C, C, A, U, G. C, 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 U, A, U. And C, G, C, G, 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 U, A, G, A, G. So hopefully I didn't make any mistake. And the last one is going to be U, A, G. Alright, so that's the 3 end. There you go. Alright, now we have the mRNA sequence. Now we can use the codon table to translate into amino acid sequence. Alright, okay. Now, first thing you need to do is to find that start codon. Remember the translation only starts at the start codon. So even though in this sequence UUG is the first codon, the ribosome is not going to recognize it because it's going to start with the start codon, which is AUG, right? So it's going to start right here. And then everything prior to AUG, nothing will happen to them. Okay, so they're not going to code, they're not going to participate, they're not going to be involved in translation. So we start here. So AUG, which one is that? Methionine, right? And the next one, CCC. Oops. So go back here. So how to read this codon table? There are 3 letters, right? So the first letter starts from the left side. So you're going to find a C here. And then the second letter is up here. So again, you're going to find a C. So C, C, that's going to be in this box. And then the third letter, you're going to look at the right-hand side. So C is here, so you can draw a line. There is CCC. So CCC is proline. Alright, okay, so CCC, AUG is done, CCC is done, and what's the next one? UAU. So again, start here, U, and the A is here, so it's definitely going to be in this box. UAU, right? U is here, so this is the first one. Tyrosine. Tyrosine, and so this is done too, and next one is CGC. So again, here C, and find G, so it's in this box. CGC, there's a line there, so arginine. So CGC is done. Okay, now GGG. G is here. G, it's in this box, so GGG, glycine. Alright, this one's done. And the next one, UAA. So UAA, right? So UAA is the stop codon. So this tells you, okay, I'm done with synthesizing the amino acid chain. Now we have reached the stop codon. So you just stop here, right? A lot of students tend to put stop here, but you shouldn't, because stop is not the name for amino acid. So you can just leave it blank. All right. So this amino acid chain has 5 amino acids. So this is the correct answer. Okay? And also we have the answers over here. So how many amino acids after translation? 1, 2, 3, 4, 5. Okay? All right. So again, the key is when the DNA template is given, make sure that if the sequence is given from, because we usually read it from left to right, right? So if it's given from 5 to 3, then you are going to have to read the sequence in the other direction, just like you are an RNA polymerase, right? So RNA polymerase reads the DNA sequence from 3 to 5. So if I give you 5' to 3' sequence, you have to read it backwards. Okay? If I give you a 3 to 5 sequence, then you can read it normally. Okay? So you can just read CTA and then figure out the complementary sequence. All right. Okay, now tRNA, so you already know that the tRNA molecules are the little workers that will kind of transport the amino acids to ribosomes, right? Because ribosomes use amino acids as raw materials to make amino acid chain. So the tRNA has a very unique structure, so it looks like this. In previous tests, I have been using an image of tRNA and asked students what molecule this is. So you need to know what tRNA looks like. It looks like a clover leaf. And you need to know there are two ends that are very important. The first end is over here. So this end is where amino acid is attached to. Okay? It's attached to. And the sequence here, if you read it this way, it's always CCA. Okay? Always CCA. The other end over here, this is the end that has the anticodon. So the anticodon is going to pair up with the sequence in mRNA. Right, the sequence in mRNA is the codon, right? So the anticodons are complementary to the codons in mRNA. So it's the same. It's like base pairing. So with codon and anticodon, tRNA knows what amino acid is supposed to be delivered here. Okay, so there's a short video about 4 minutes. You can read. It's an animation on how translation works. Okay, so it's pretty interesting. So again, just like transcription, you have 3 steps: initiation, elongation, the chain is growing, and then termination. That's when translation ends. Okay, so just real quick, you need to remember that ribosomes have 2 subunits. So there's a small subunit and there's a large subunit. So the small subunit is going to bind to mRNA first. Okay, so it's going to locate where the start codon is. Right? And then the tRNA is going to deliver the methionine over here, because the anticodon over here, UAG, pairs or is complementary to AUG, the start codon. So this ensures that the tRNAs can deliver the correct amino acids. So once the first amino acids in, then the large subunit will also come over, and now they're ready to add the rest of the amino acids. Okay, so this is the initiation, and then elongation. So in ribosomes, you have 3 spots. So you have A, P, and E. So the A is for amino acids, P for polypeptide, because when you connect the 2 amino acids together, you are building a polypeptide chain, right, to link the 2 amino acids. And then the E, you can remember, it's for exit. So the second amino acid is going to be delivered and it's going to enter this A spot. Okay? And then there's the polypeptide chain forming between methionine, the first amino acid, and the second amino acid. And then next you're going to move on to the E, to the exit, and then so basically just kind of moving forward, right, until the tRNA gets to the E spot, so it's going to exit the ribosomes. But these 2 amino acids have been linked. Okay? So again, the third amino acid will be delivered over here, so the tRNA is going to enter A, and then the second amino acid will form the polypeptide chain with the third amino acid. So now you have a 3 amino acid chain. So that's elongation. And one mRNA can have multiple ribosomes attached to it and make them the polypeptide chain. So this is what this picture is showing. So you have one mRNA, especially for eukaryotes, the mRNA can be very long, right? And you don't want to just have one ribosome that scans through the mRNA because that's very time consuming. So what we do is a very efficient system. So we're going to have multiple ribosomes attached to the mRNA, and each one will start making the amino acid. So that's going to be really, really fast, right? You have multiple production lines going on at the same time.